Hey guys, Kenna here. So today we're going to go ahead and talk about meiosis. Now, if you remember what we talked about before with respect to mitosis, a lot of things here are very similar. There are some key differences, however, I'm going to have you watch out for. And one of the big ones is that meiosis, unlike mitosis, is what we call reduction division. And what that means is that we're going down to half as many chromosomes. The goal in mitosis was to produce two identical daughter cells, but in this case, we're going to produce four haploid, and we'll talk about what that term means, gametes or sex cells. So this is the sperm and the egg. All right, so let's go ahead and jump right in and let's take a look at our essential questions for today. Number one, what is the purpose of meiosis? I kind of gave that away a little early, but it's important to kind of keep in mind what the goal is of mitosis versus the goal of meiosis. They're similar, but they have a very different goal in mind, and as such, there are some differences in the process of how we get there. Number two, how is meiosis different and similar to mitosis? Make sure that if you're trying to answer this question, you include the terms diploid and haploid in your answers. And number three, what is a crossover and how does that relate to meiosis, both one and meiosis two? Now, we talked about mutations last class, and so we're going to talk a little bit more about mutations today and next class, and that'll help us get a better idea of how these changes come about and the variation that exists within a population of a specific species. So let's begin by going back to mitosis a little bit and review the stages of the cell cycle. So last semester, we went over the stages of the cell cycle, G1, S, G2, and M. So let's start with G1. What happens during G1? Hopefully you said the cell grows, and this is where cell growth happens. What about in the S phase? Hopefully you're able to come up with this is where DNA replication takes place, what we were talking about a couple classes ago. Now, what happens during G2 phase? That's right, prepares for mitosis, and then M phase is mitosis itself. When we look at mitosis, there are four phases of mitosis or stages: prophase, metaphase, anaphase, and telophase. In prophase, the cells are going to go ahead and condense their DNA from chromatin into chromosomes. We're going to dissolve the nucleolus and the nuclear membrane. We're going to form the spindle fibers, and we're going to go ahead and make sure we move the centrioles to opposite sides of the cell if it's an animal cell. Remember, plant cells don't have centrioles. Then in metaphase, the spindle fibers are going to pull the chromosomes, which are now attached to the spindle fibers, to the center of the cell. M meta, M middle. Okay, that's the best way to remember it. In metaphase, all of the chromosomes are lined up in the middle. In anaphase, they're going to go ahead and separate the two sister chromatids and pull them to opposite sides of the cell. Once they get to the opposite ends of the cell, they're going to go ahead and ball up and then unwind again, and this is going to be in telophase. But also during telophase, while we're unwinding the chromosomes into chromatin, we're also reforming the nuclear membrane and the nucleolus. Because remember, mitosis is the division of the nucleus. Cytokinesis is the division of the cytoplasm. Here's another example of the same thing. There you go. You can see G1 is cell growth, S phase is DNA replication, G2 is prep for mitosis, and there is M phase with mitosis and cytokinesis, the actual shortest part of the cell cycle, cell division. And we encourage you to go ahead and remember these different phases of M phase or stages of M phase using the acronym PMAT. I throw in IPMAT here because interphase is that 3, G1, S, and G2 that comes before it, and it must go through interphase before it can go ahead and go through PMAT again. Prophase, metaphase, anaphase, and telophase. Okay, so if we have that fresh in our mind, let's start looking at meiosis through the lens of mitosis. There are many similarities between mitosis that we learned last semester and meiosis that we'll cover today. To start with, the stages are still prophase, metaphase, anaphase, and telophase, but with a twist. and cytokinesis still takes place too. The big twist? We're going to do it twice. Let's look closer at the process of meiosis and then compare the two processes. Now, when we finished going over mitosis, we gave you some chromosome numbers to look at. Chromosome numbers vary considerably among organisms. These numbers may differ a lot even between closely related species. You see that the human has 46 chromosomes. That's one of the ones I highlighted last time to kind of keep track of. But that doesn't mean that the goldfish with its 94 chromosomes is somehow more complex or more evolved than we see with the human. It's simply how the genes are organized into separate chromosomes. We also pointed out that there are some species that have different numbers, whether it's the male versus the female, like we see in the honeybee, or when you start looking at the orange, where you have two different species, an 18 and a 36, and then you have hybrids, which have one sex cell from each at 27. When you look at those numbers, hopefully you were able to notice that they were all even numbers, divisible by 2, and this is because all of the species listed on the previous table reproduce sexually, meaning that they have two parents from which they get one copy of each gene, for a total of 2 copies of every gene. These two sets of chromosomes are what we call homologous. Remember that prefix homo means same. So homologous means that each chromosome came from the male parent, has a matching chromosome that came from the female parent. That's what we would call diploid, or the 2n number, 2 times one set of chromosomes. A cell that contains both sets of homologous chromosomes, as I mentioned, is diploid, meaning 2 sets. This number of chromosomes is often written as 2n. The sex cells, or gametes, this is the sperm and the egg, remember, of sexually reproducing organisms are said to be haploid, 1 set, and this number of chromosomes is often written with the letter n. So you can see that the diploid stage has twice as many chromosomes as the haploid stage. Let's go ahead and give you an example so it's more clear. The adult fruit fly, Drosophila melanogaster, has 8 chromosomes. Thus, the diploid number is 2n is 8. Now, if you can do some simple manipulation in terms of your algebra, we can go ahead and divide both sides by 2, and we get n equals 4. So the gametes, or sex cells, for each fruit fly is then going to be 4 chromosomes. You get a sperm from one fruit fly and an egg from another fruit fly. 4 plus 4 brings us back up to the diploid number of 8. Because we have to cut the number in half so that we can go ahead and have this recombination through fertilization, meiosis has to be used. And so instead of doing just a straight copy and division like we do in mitosis to produce 2 identical cells, we do meiosis to produce our gametes. Again, let me highlight this. This is a key piece here, but meiosis is what we call reduction division. Okay, reduction division. We go from our original number of chromosomes down to half that number. If we're going to go ahead and look at humans, for example, you'll remember that humans have 46 chromosomes. Well, what that means is that they actually have 23 chromosomes from mom and 23 chromosomes from dad. Now, if we do a little simple math, you can see 23 plus 23 is our total of 46. For humans, 46 is the diploid number, and for humans, the haploid number for sex cells is 23. So we'll put up here diploid, and down here we'll go ahead and put haploid. Okay, so it's important to keep those two straight in your mind because that's going to be important in terms of sexual reproduction and fertilization. Okay, so let's clear that and go ahead and move on. We'll break meiosis down into 2 parts. The key to understanding it is to realize that the first half of the process is incredibly similar to mitosis, except for the fact that we line chromosomes up in homologous pairs in what we call a tetrad. In a tetrad, there are 4 chromatids versus the 2 paired in mitosis, because we have both the maternal and paternal chromosomes for the same chromosome lined up side by side. During this pairing, sometimes we actually get what's known as crossing over. So if we go ahead and have the two coming together as we go into late prophase, they end up pulling side by side and the spindles attached to them so that we can pull them to the center of the cell in metaphase. Now, when this happens, sometimes the inside legs of the chromosomes get crossed up. And when that happens, they get tangled. Now, the best analogy I can give you for this is I want you to think of a slinky. How many of you have had a slinky before? Have you ever gotten a slinky tangled? How easy is it to get them apart once they get tangled? Now, hopefully, you think about it and you realize, yeah, it's pretty hard to get apart a tangled slinky. But the only easy way to get them separated is to cut them. And so this is what your body's going to go ahead and do, is it's going to get tangled, and then it's going to go ahead and cut the tangle and try and put the pieces back where they belong. But sometimes it doesn't put them back in the right chromosome. And so you end up swapping genetic information from one chromatid to another. And this produces a new combination of alleles. So let's take a closer look at this process of crossing over. Higher organisms, such as plants and animals, reproduce through sexual reproduction. A male, a female, and those are going to reunite those gametes to produce the diploid number. The offspring possesses a combination then of characteristics from both the mom, maternal, and from the dad, paternal. When we start looking at these chromosomes, they're going to go ahead and pair together in meiosis, and genetic material sometimes is exchanged because of this process known as crossing over. Crossing over can only occur when homologous chromosomes synapse come together side by side. So there's some key terms here when we go ahead and start looking at this. Here is our maternal chromosome. So you can see that we've replicated it. We've already gone through S phase. Okay. And we have wound it up into our little X's that we recognize as chromosomes. So 2 chromatids making up a chromosome, and this is your maternal. We go ahead and have the paternal, oops, too fast. The paternal here, which is going ahead and looking at the same sort of thing. 2 chromatids make up the chromosome. This is the father's paternal. And so lining up side by side like this is what's known as synapse. So let's go ahead and highlight that really quickly. This term right here, synapse. Synapse is the position of them lining up side by side. Okay, so synapse, we can go like this. 2 homologous chromosomes line up side by side. Okay, so that's what synapse is, and we have to kind of keep that in mind. We want to keep that in mind. All right. Now, before you go ahead and start moving too far on, you may or may not have seen actual photographs of chromosomes. This is an electron microscope image of chromosomes. You see here the X and the Y, and we'll talk a little bit more later about why there's such a disparaging difference between the size of the two. Turns out that the Y chromosome has actually lost a whole bunch of genetic information. This is the configuration for a male in mammals, a 1X. Everybody has X's, whether they're male or female, but the Y is only found in males, and it's missing all this bottom information here, and that has some implications, and we'll start looking at sex linkage later on when we talk about genetics. Here, what I have the pictures there for is to go ahead and show you this tightly wound structure that has all the histone proteins that are coiling and supercoiling up the chromosome to make these little X's. So it really does, in reality, look kind of like a slinky, which is why they do get tangled when they're moving about in late prophase. And we have to fix that. They can't be tangled if we're going to separate them as we start going into metaphase into anaphase. Okay. And so think about that and think about slinkies and how difficult it is to separate slinkies. Okay. So let's clear our notes here and go ahead and move on to the next slide. All right, so let's continue with crossing over. Now, the point at which a crossover occurs is known as a chiasma. Okay, so if we're going to go ahead and highlight this in terms of this point right here, where you can see this maternal chromatid overlapping with this paternal chromatid, and they get tangled. This is that tangle spot for the slinky we talked about. That's what's known as the chiasma. Okay. All right. So chiasma is in the process of forming, and in this case, equivalent sections of the chromosome can be exchanged. Now, it's not always equivalent. Sometimes they exchange in an uneven amount depending on how the overlap occurs and where it ends up going to. So, as we start looking at this, hopefully we begin to realize that once it gets corrected, let's say another enzyme comes in and says, oh, there's an error, cuts it, and tries to replace it, sometimes it puts it back in the wrong spot. And when this happens, you now have a new combination of chromosomes that didn't exist before. Okay, there's that crossover point. There's the chiasma where that crossover point occurred. And each of the two homologous chromosomes has now exchanged some genes between the neighboring chromatid and leaving the outer chromatids untouched. So again, notice that the top and bottom chromatids are unchanged. It's only the inside chromatids, the ones that were lined up next to each other, which have swapped information and become new combinations. So there's your mixed types, there's your original types, and this is what's then going to go ahead and pass on to the intermediate cells of meiosis I and eventually on into the sex cells or gametes. And this is one of our forms of mutation that's going to produce new combinations in the offspring. Now, during the final division of meiosis, the chromatids that were bound together are going to separate. Okay, so let's assume that we have a cell membrane that forms between, and we now have two completely separate homologous pairs, but they're no longer identical because of the crossover. These are then going to divide again, and eventually this will go ahead and produce 4 chromatids with recombined genes in 2 of them for a total of 4 genetically different gametes. Now, if some of them were not involved with the crossover, like these first 2 you see here, they look the same as they did before the crossover happened. But those that do have the crossover, you'll see here, the wild types, okay? These mixed types end up producing new combinations that neither the parent had. Okay, let's go over the process one more time. So we have this setup. Can you guys remember what we call it when we have 2 of the double sets of chromosomes lined up side by side? Pause the video, go back through your notes, see if you can remember. Hopefully you were able to come up with the fact that these are homologous chromosomes and they are lined up in the position of synapse. Okay, so we can go ahead and write that here just to kind of remind ourselves. We have homologous chromosomes and they are in the position of synapse. And when they line up in synapse, they can go ahead and produce a point of entanglement, let's call it, okay, where one of the chromatids on the inside, and it's the ones on the inside, gets caught on one of the chromatids on the inside of the other pair. Does anybody remember where this point is called? No? Okay. So when we go ahead and start looking at it, this point where the 2 overlap is called a chiasma. Chiasma equals the point of crossover. And Well, hopefully you remember that the body doesn't like this, and so it wants to fix it. And so what it does is it brings in an enzyme. Again, I'm not going to give you the name here, but this enzyme is going to actually come through and cut the two apart so they're no longer tangled. Think about our slinky that got hooked together. And when it does that, it tries to reattach them back where they belong. And when it does so, sometimes they exchange genetic information. And these are now two mixed types of chromatids in our homologous pair. Okay, so that's really what crossing over is in this late prophase stage. As we start beginning to move towards metaphase, they get pulled together and the homologous pair exchanges genetic information on the inner chromatids. Okay, all right, so let's go ahead and take a look. Oops, let me put that back for you. Let's go ahead and take a look at the whole process of meiosis, keeping in mind what we've discussed in terms of mitosis before. So we begin at interphase. This part is the same. The cell's going to go ahead and grow. It's going to go through S phase where it replicates its DNA. So if it was a Drosophila melanogaster, for example, who has 8 chromosomes, by the time we get to the end of S phase, now we have 16. Then we're going to go ahead and start going through division. We'll go for Prophase I, Metaphase I, Anaphase I, and it is important that you include that one label. Because if you don't, how are we supposed to know if we're in the first half of meiosis or in the second half of meiosis? Now, it does go into telophase I, but I don't think it completes the process in the same way you're thinking, because as it begins to finish the division of the nucleus, it immediately starts to go into meiosis II. So we can include telophase I here, but it's not complete. Same thing with cytokinesis I. We're going to go right into meiosis II. And in this case, the 2 cells produced by meiosis I are going to go right into a second meiotic division. And here's the key. They go into that second meiotic division, but they do not go through interphase again. Let me repeat that. They do not go through interphase again. And as such, you don't duplicate the chromosomes again. All right, let's go ahead and make a note of that right here. Okay. There is no interphase between meiosis I and meiosis II. This is really important. So once we've done one replication in S phase, we do one division and then immediately start on another division, which is why it's called reduction division, because the number of chromosomes goes down. Okay, no interphase between meiosis I and meiosis II. Okay, so that being said, let's go ahead and continue on and look at meiosis II. You have prophase II, so we're going to do that whole condensing process again. We're going to go ahead and build up the spindle. We're going to go ahead and move the centrioles. Everything we talked about in mitosis. What happens really is the same. We go through metaphase II, anaphase II, they're pulling apart, and then you go into telophase II, and we go ahead and produce 4 haploid, in this case, daughter cells. 4 haploid. We have now cut the chromosome number in half. Now, it'd be nice if this was just the end of it and we were finished and all is done, but there's one more little piece we need to cover. This process is slightly different between males and females. While both males and females produce 4 genetically different haploid cells, all 4 of them in men become sperm. But in females, one of them ends up with all of the cytoplasm and all of the organelles in it. This becomes the egg, and the other 3 are tiny little sacs of leftover genetic material we call polar bodies. And these are not usually involved in reproduction. Here's a visual representation of exactly that process. You'll see we start out with the original cell here on the far left. We go through prophase I. Notice we have our 4 chromatids. So there's our tetrad in the position of synapse. Okay. It's going to go ahead and line up as it comes towards the middle. We move from Prophase I to Metaphase I. At that point, it will have corrected any crossovers if they happened. And so then we're ready to go into Anaphase I. We finish Anaphase I, and as we start to go ahead and separate in telophase cytokinesis 1, we immediately start into Prophase II. And Prophase II is going to go through these same processes, but with each of the other cells. We'll go through Anaphase II, which eventually leads to our 4 gametes. Now, again, in spermatogenesis, the process of producing sperm, all 4 of those are going to be modified and become sperm cells, haploid sperm cells. But if you take a look at the female here, you'll notice that there's 1 large egg and those 3 small polar bodies, which are typically recycled. This is the one that has the ability to be fertilized. Still haploid, but this is why these can be so small, the sperm cells can be so small, is that the oocyte that formed it, most of the cytoplasm and most of the organelles end up inside the egg cell. All right, let's do a quick review of mitosis versus meiosis. This would be a great table for you to go ahead and make sure you take down in your notes so that you can go ahead and compare the 2 processes, because there's a lot the same, but there's some key differences. Number 1, let's start off with the goal. In mitosis, our goal was to produce 2 genetically identical cells, okay? Identical daughter cells. But in meiosis, we don't want to produce 2 identical cells. Instead, we're going to go ahead and produce 4 genetically different sex cells. This is your egg or sperm, depending on if you're male or female. We go through mitosis and we do cell division 1 time. So we do 1 S phase, so DNA replication, and 1 division. So we go from 46, our original number, all right, to 92, and back down to 46. So each cell is exactly the same as the parent cell that we started with. But in meiosis, we're going to divide twice. We still start out with our 46 if you're human. We're going to go ahead and copy that up to 92. Then we'll divide down to 46. But instead of going into interphase here, we're going to immediately start to divide again and go from 46 down to 23. So you see in outcomes, mitosis produces 2 identical cells and meiosis produces 4 genetically different sex cells. Okay, inside those cells, at the end of mitosis, you end up with all 46 chromosomes. This is a diploid cell, and in meiosis, we only have 23 or half that number. This is the haploid cell. Okay, let's go ahead and put that label here just to remind ourselves. They have diploid. This is what we have when we do mitosis. And then over here, we're going to have haploid. This is what we get when we're done with meiosis. Remember, it's reduction division. We're taking that original number of 46 and we're cutting it in half. Okay, guys, that's the big things I want you to take away from this today. We have a good activity for you, and take care of yourself. Stay healthy, stay safe, and I'll talk to you soon. Take care of yourselves. Bye-bye.